Hello and welcome. You are listening to a podcast by the Milner Centre for Evolution at the University of Bath. I'm Professor Turi King, your host, and today I'm talking to Guillaume Méric, an Associate Professor whose research focuses on the human microbiome. Guillaume, for those who don't know, what is the microbiome?
So we as humans have co-evolved with the microbial communities that colonize us, and probably from even before we were human. And this has resulted in a complex assembly of trillions of microbial cells from thousands of species that are inhabiting on or in our body. The gut is a particularly heavily colonized body site of ours. Microbes there fulfill very important role for our health. They help digest our food, they produce vitamins, they interact with our immune system, and they protect us against a whole range of gastrointestinal pathogenic infection. And they even communicate with different organs, which can include our brain. Our gut is actually our largest immune organ. About 80% of the body's immune cells are there, partly because of the constant negotiation that our body has with these trillions of microbial organisms. And in the last few decades, researchers have found and characterized that disruptions in the balance of this microbiota seem to be linked to a wide range of diseases and health conditions.
So let's go back to this a little bit, because people are really starting to clock just how important the microbiome is. So you've got like these trillions of cells, haven't you, in your gut? What are they doing in there?
A lot of them are there since our birth. There is debate to know whether they are there before our birth as well. But there's a lot of various different species that are inherited from our mothers, that are transmitted vertically, as we say. Those are very interesting in themselves because we have a sort of inkling that they are more linked to our genetics and our ancestry and our population we belong to. And there are others that are also coming from external input, such as, diet or exposure to various things. Underpinning what the bacteria are doing is a very difficult task. There is a sort of consensus now that the bacteria that are producing good things for our health seem to be producing some small molecule metabolites that are called short-chain fatty acids. And those have been linked in a lot of population studies and population health cohorts with positive health outcome and notably with positive cardiovascular health outcomes.
And we all have our own personal microbiomes that are the product of, as you say, so some coming down to us vertically from, birth, ancestry, and other bits from what we're eating. So we all presumably have our own personalized microbiomes. And how do they help us in terms of health? Because you've just been mentioning about cardiovascular disease. How do they affect our health?
So how they affect our health is not very much known. Aside from this production of short-chain fatty acid that I mentioned, the mechanisms are a bit more elusive. And at the moment, what is being done is just to try and robustly associate particular taxa, particular species, particular functions with health outcomes and with positive or negative health outcomes. And the reason why it's difficult to pinpoint these things down is exactly what you said. It's that it's immensely variable in people. You have commonalities in different people. We will be colonized, for instance, the both of us, by the same species of bacteria, perhaps different strains of this bacteria, but we will have the same species. And normally speaking, the dominant bacteria are roughly the same if you just look at what's there. The quantities might vary, and also the dynamic of these different interactions between these bacteria might vary. And sometimes, depending on the different conditions of health, depending on your culture, depending on your diet, depending on your genetics, you will have sets of important bacteria that are different from each other. And so this individuality in the microbiota profiles is something that makes it very difficult to know what's good, what's not in people. But it's also making a sort of population scale approach of research have more sense, because if you look at a lot of different people, if you try to control for various different factors like this, you can try to compare things and, find commonalities or things that are different between people.
And that's basically what you're trying to do. So how do you do that? How do you tackle this huge question as to what's going on?
It's not easy, but it's exciting. So my group is at the intersection of various different domains. I'm a microbiologist myself, but the work we do is at the intersection of population health, of clinical microbiology, bioinformatics, and epidemiology. The goal of our research is to understand how the human microbiome interacts with health, and particularly, when I was mentioning different diseases, particularly with non-communicable disease like heart disease, diabetes, or more recently, liver disease for us. So the way that we do this is, in my group, entirely computational. We use large-scale, secured, anonymized human population cohort datasets that are including a microbial component. So that would typically be metagenomics datasets from the gut. We sequence everything we can find from the gut. We try to assign what is there, in terms of bacteria, and we analyze this. We work as part of consortia, big collaborations with Nordic population cohorts, mainly from Sweden and Finland, but also other locations. One of the interesting aspects of these Nordic cohorts is that they are extremely well phenotyped. We have lots of matching data for every participant that is enrolled in the cohort, whether in the form of questionnaire data, food habits or lifestyle habits, but also prescription data, adverse health outcomes, electronic health records. Everything is matched for these participants. And even more interesting is that for one of our cohorts of interest that is called FinRisk from Finland, we have a very long follow-up. We have up to 25 years of follow-up after the sampling of the microbiota in the people. So we know what happened to these people. We gave a sample of their poop in 2002, and we can use this formidable wealth of data to sort of look at possible links between the gut microbiota and future health. And so the type of research we have is very generalistic and very generalist, but uses this sort of framework to look at various aspects of health and the microbiota.
But that's really cool, because presumably what you can do is, because you've got these long-term studies, you can go, well, these people had these particular bacteria, and they were more likely to go on to develop this particular condition versus these people. And are you in the early stages of trying to understand that, I'm guessing? Because this is a relatively new field, isn't it?
It is one of the things that is actually very exciting about it. I find that the field is renewing itself really quite a lot. There's a lot of technological enhancements or novelties that have arrived in the last decade, I would say, that make things maybe not conceptually very new, but much more upscalable. You can start looking at very large sample sizes. You can start looking more in depth in those samples as well. And this allows, not to reinvent the wheel, but to confirm a lot of things that we could have inferred from smaller sample sizes, from smaller groups of people. But if you contextualize them in the context of populations, that's when you can start having, these findings, basically. So this is a bit why it feels like we are at the beginning all the time, because we are constantly refining what we suspect and what we have seen with low statistical power, low sample size, but we are confirming it or infirming it in various different populations, various different contexts. The more this is done, and I was talking about Nordic populations now, they were the first, the Nordic countries were the first to establish very good electronic health records and very good sampling of populations. This is why we are working with them. But of course, now other countries are starting to do this. And the more we do this, the more we try to represent human populations as a whole, the more we see the differences, the more we see that it is a complex thing. And of course, when you talk about countries like the UK, countries where there's a lot of people, the population is diverse, there's many different sorts of microbiota in there, you can see the challenge of making generalistic statements about health. It is difficult still at this stage to link anything that the microbiota is doing generally in a population. But we are having quite a lot of ideas now. We're starting to have a good picture of it.
So I was going to ask you that. Are you seeing any trends yet? Or is it still really early in the research and you're not seeing anything particularly at the moment?
So it's very hard to say anything very generally speaking, in the gut microbiota field, because there's so much of this variability. However, there seem to be some strong facts that are emerging, which is, I was mentioning earlier, those bacteria that are able to produce short-chain fatty acids, that are small metabolites that have been linked to a lot of positive health outcomes. Those bacteria are very diverse. They come from a very sort of different taxonomies. They have the phylogenetic diversity. It's not just one species. It's not just a group of species. There's quite a lot of them. The trend that has been shown is that they seem to be overrepresented. They seem to be growing in our gut when we have a diverse diet that contains a lot of fiber. That's why you hear about eating a lot of fibers, and fiber is good for you, and particularly different kinds of fiber, not just one kind of fiber. The more you can, the better. But this is something that has been shown to promote those short-chain fatty acid producing bacteria with positive cardiovascular health outcome. One interesting aspect as well is fermented food. Fermented food have been shown not only to contain a lot of short-chain fatty acids themselves, but also to have those bacteria that are producing quite a lot of short-chain fatty acids. So generally having a balanced diet full of fiber, possibly with fermented foods of many kinds, kimchi, sauerkraut, stuff like that. This is, we think, the link between health and the gut microbiota at the moment.
And this is where the Western diet is terrible, isn't it? Because this is all about how different cultures will have different diets, which will have more or less fiber in. And you hear this a lot at the moment, this drive.
Generally speaking, I don't know if I'm a fan of calling it the Western diet, because I find that it's a bit confusing. What I prefer to say is that foods that would contain a lot of ultra-processed ingredients, typically, if you look at, a pack of triangle sandwich, that you can buy, and you look at the ingredients behind it, of course, there's a lot of ingredients in that sandwich, but there's a lot of ingredients as well that will not be food, that will just be preservatives. We sort of inherently know that it's potentially probably bad for you, but we don't really know why. And there has been research for this, and notably research into aging, not necessarily so much with the gut microbiota, but research on healthy aging, for instance, has shown that those particular consumptions of ultra-processed foods seem to be very bad. It seems to be something that is associated with a worse aging than if you have raw food, if you have fiber food and things like this. So I wouldn't be surprised also that the microbiota is affected by this. So it's better to think about how much processing goes into your food and how closer you are to the fresh product or not, really.
So one of the things you mentioned early on is that it's not just the microbiome that you've got, but how it interacts with your own genome. So this is something that you're looking at, isn't it?
Yeah. We've had some effort trying to look at this ourselves and also in collaboration. So we were part of a collaborative work recently that was published in Nature Genetics, and this was led by the same Swedish colleagues that I was mentioning about, on the links between human genetic variation and the gut microbial variation. So that study was the largest of its kind to date, and it was the sort of prolongation also of another microbiome GWAS study we have led in 2022 when I was in Australia. And we were looking at roughly the same things in different settings, but those results in 2026 were more detailed and consistent. So what did we do there? What we did was we asked, does your DNA and do your genes influence which bacteria live in your gut? We scanned the entire genome of over 16,000 people, and we compared it to what bacteria were found in their gut. We found 15 robust genetic associations, so it's specific spots in the genome that are reliably linked to having more or less of a particular species of a bacterium. So there were some expected results from past studies. There is a well-known association between LCT, or the lactase gene, which governs whether adults can digest milk, and the species Bifidobacterium. We also found specific bacteria linked to the ABO locus, which is governing blood type, which is also sort of known, and it was fascinating in itself. But we also found new ones that were quite interesting. There was a sort of discovery that I found particularly elegant. So we found variants that were near a pair of genes that encode receptors for those short-chain fatty acids that I was mentioning earlier. So they are molecules that the gut bacteria produce themselves, and they were linked to how diverse the gut microbiome is overall. So our genome appears to contain the sensors that are talking to our bacteria like this. There were other findings. We found the connections to genes that are involved in the mucus layer composition. So our gut epithelium is covered with mucus, also with bile acid production or hormone signaling, a lot of various different things. The takeaway message from these studies and this work that we're doing is that the gut environment that our own genome creates, the sort of micro changes in the environment that our own genome creates, can shape which bacteria can thrive there. And we are only starting to understand it for the reasons that we mentioned about sample size and technologies. So yeah, it's exciting work there.
But that's really interesting, because if it's interacting with your own personal genome, is the idea that eventually that could be used in a sort of personalized medicine sort of way? Is that the ultimate aim?
It is the goal. The goal is to understand the diversity in humans, the diversity in their genes, the diversity in their disease risk, the diversity in their disease history, maybe, in their medication history. As we say, it can have an impact. But if you add on to this the complexity of the gut microbiota, which is very much influenced by diet and what you eat and who you meet and what you do, it becomes extremely complex. So one of the ways that is very good and seems to be one of the most promising way to shed some light on this is to do large population studies, to try and see in a large number of people what commonalities can we see. What can we see when we make groups of people and we compare them, all else being equal? What we can do as well is to organize clinical trials where we are trying to investigate the function of those particular microbes. When some people have particular microbes or when they are given particular microbes, as in probiotics, for instance, but not just, what is the effect? Can we measure this effect? One important thing that is deriving from more controlled studies like in vivo studies or clinical trial studies is that we can have a sense about causality. Causality is the elephant in the room in every microbiome study that you see. Basically, when you link a particular microbiome state or particular microbes to a particular disease, you have no idea whether the microbe is causing the disease or the disease is causing the differences in the microbiota that you see. We can see a lot of these associations. You can make correlations and you can see that things are associated. Trying to derive meaning, trying to derive useful information from this goes far beyond the correlation work that we are starting to do. You need to go through the process of doing smaller scale studies, to do them in vitro, in the lab, in animal models, in patients with clinical trials. It takes a long time to derive meaning from microbiome studies, but we're getting there.
So what's next for you?
So there are many things, as you can imagine, that we can look at, but there's a particular focus that is coming to fruition for us and that is kind of close to my heart. We have been looking at a particular fraction of the human gut microbiome, which is represented by archaea. So archaea are a totally different type of organisms. They are not bacteria, they are not eukaryotes, they are completely a third domain of life. And in our body, they are mainly thought to be linked to methanogenesis. They produce methane. So what they do, basically, is that they associate very intimately with these bacteria that we were talking about that are consuming fiber from the diet. So bacteria that ferment fiber will produce all of the molecular components that are required for these archaea to produce the methane. And there is an interesting thing to look at there because of this metabolic interaction, we can have very, very, very robust associations and co-abundance association between particular archaea and particular bacteria. We have these sort of co-abundance networks, as we call them. And what is very interesting is that until now, archaea had been very disregarded in, probably any field of science, mainly because it was very difficult to study them. It's very hard to culture them. We are collaborating for this with a lab in Austria that is led by Professor Moissl-Eichinger, and they are the best lab working on human archaea in the world. They have the way to culture these archaea. These archaea grow in total anaerobiosis. They grow with hydrogen. You have to inject hydrogen. So imagine having canisters of hydrogen in the lab. This is not really 21st century science, but they have all of this set up. And they manage to grow these archaea. And they have invaluable knowledge for this. And we're about to publish a paper on this. And we think it's a very exciting sort of first foray into the globality of the archaeome. Basically, one of the results that we find is that humans seem to be colonized by different groups of archaea with different potentially health associations. But this is all very exciting stuff, and I hope that we will be able to develop that further in the future.
Guillaume, thank you so much for talking with me. This was a podcast by the Milner Centre for Evolution at the University of Bath. I'm Turi King, and thank you for listening. If you have any thoughts or comments on this or any other episodes, please contact us via our social media. For more information about the Milner Centre for Evolution, you can visit our website.